Thank you very much. So I firstly want to just thank the organizers of the event for inviting me to present. And I do send my apologies that I can't attend in person, but really pleased to be able to showcase some of the work that we're doing here in Manchester Metropolitan University. I'm going to focus on the assessment of brain creatine and just speak a bit about the different considerations that we should be having when we're going down this route. Just before we start, I have no disclosures to declare, but what I did want to do is just acknowledge the other people who are involved in this work. This is very much so a team effort. So I think before we get into any of the data or go any further, I'd just like to thank the other members of our team who are contributing to this work. So in terms of what we'll talk about today, just a bit of a session overview. I'm going to start by just giving us some background or laying a bit of foundation towards why actually assessing and potentially increasing brain creatine might be useful in some different settings. Then we'll move on to focus on how creatine is actually measured in the brain. So we'll spend a good bit of time on that. I also wanted to look at just bringing your attention to some of the existing reliability and repeatability data on brain creatine assessment. And we'll speak a bit about this in the context of existing creatine supplementation studies that have sought to change and manipulate brain creatine concentrations through oral supplementation. And then I suppose the bulk of the talk will focus on how we can then go and optimize the brain creatine measurement. We'll speak a bit about different factors that can potentially affect brain creatine and kind of build on the concept that we need to try and control for these factors until we better understand how it might vary. And then the final aspect is just a bit of data that we're working on at the minute in terms of the effects of an acute bout of exercise on brain creatine. So firstly, just a bit of background. I suppose it's fair to say that creatine is most extensively researched historically in the context of skeletal muscle, but there is definitely an emerging kind of body of literature to show that creatine has applications to a range of different organs and different phenotypes. And I suppose one of these that has really come to light is the potential role that creatine has in brain health and our cognitive function. And I suppose the mechanism through which creatine has been proposed to support our brain health is in this whole energy supply chain. So you can see in that illustration that Professor Darren Kando nicely shows is the potential role that creatine supplementation might have on improving the energy availability in the brain. And it comes down to really the reversible reaction whereby creatine or phosphocreatine specifically in the way it's stored in the body and in the brain donates that phosphate to ADP to provide that energy. And it has been proposed that creatine supplementation and creatine in the brain is particularly important for any phenotypes that has a significant metabolic demand. And this could be chronic cases such as aging or disease, or it could be acute increases in that metabolic demand, such as brain injury. And then it's believed that the creatine concentrations in brain are lower in these different cases of poor cognition and brain health. Again, brought about through old age or several different diseases. And I suppose it stands to reason that if we could potentially increase our creatine stores in our brain, we might be able to support that energy demand in these different contexts. And it would be a very widely accessible way of doing that. So just kind of to show a few examples of the actual applications that assessing brain creatine concentrations and potentially manipulating this brain creatine dynamics have. I just wanted to start by briefly giving you some examples with the kind of public health perspective. And I guess the promising thing is the role that creatine has in energy provision and neuroinflammation. It is quite apparent that it could have potentially quite profound implications for a lot of different phenotypes that are characterized by these three different themes as such, which is the dysfunctional energy processing, neuroinflammation, and reduced oxygen availability. And so if we can assess brain creatine dynamics and potentially manipulate those dynamics and improve the availability of creatine and potentially energy in the brain, we might be able to help with these different phenotypes. So aging is perhaps one of the biggest areas in which creatine supplementation has been proposed to benefit our brain health. And it's well established that changes in our brain energy metabolism often lead to this structural degradation in our brain tissue, and then this will then manifest as an impaired cognition. And so if we can potentially increase our creatine stores, we might be able to help combat or ameliorate some of this typical age-related decline in cognition. With stroke, this is a phenotype that we know that's creatine supplementation before a brain ischemia will actually mitigate some of that resulting damage from that event. And there are kind of this hypothesis, I suppose, a working hypothesis, is we're yet to really show this in humans, is that if we can acutely supplement with creatine following a stroke, we might be able to benefit that patient recovery in those settings by, again, trying to restore the kind of usual energy metabolism in the brain. And then neurological disease is another big theme where creatine has been proposed to have quite promising applications. And it is often the case that we do, again, get this impairment in our brain energy metabolism that precedes the manifestation of these diseases. And there are certainly evidence, largely from animal models, that shows that creatine supplementation can help with some of these disease-specific markers, and in the case of Alzheimer's, that would be such as the amyloid beta or tau tangles. But again, it's largely based upon animal models. If we just shift slightly for anyone in the audiences that are interested in sport or that performance setting, one big theme that creatine has been proposed to support or to have some benefit is in the area of mild traumatic brain injury. And again, comes down to the kind of common pathways that creatine and the neuropathology of MTBI share. And there have certainly been some reports of reduced brain creatine concentrations in those that have just suffered from an MTBI. And there are also some data from America that show in American football athletes that the creatine concentrations actually reduce across the American football season, even in the absence of a official concussive event, showing that the kind of impact, even if it is subconcussive level, it will have some impact on the brain creatine stores. So again, it gives us this working hypothesis that if we can potentially get in there with supplementation and increase brain creatine in the brain, we might be able to support the brain health of these athletes. And then something that I'm quite interested here as well, and something that we're working on, is the potential benefits of creatine supplementation for cognitive function under fatigue. And there are quite interesting data coming from sleep deprivation work that shows that acute dosage of creatine can help to support our cognitive function in these sleep deprived and fatigued states. But what we really don't know is how the concentrations of creatine in the brain actually acutely and transiently perhaps change in response to these cognitive challenges. So there is really a lot of scope for us to better understand the brain creatine dynamics in the context of supplementation and the potential benefits of our decision making and our cognitive function when we're in this fatigued state. But I suppose, something that we really need to hammer home is that we need more original human data. So just with a quick PubMed search, the amount of papers that have been published in the last 10 years is around 100 papers, but the vast majority have been review articles. And so although we have this, really nicely accumulating body of evidence supporting creatine supplementation and benefits for brain health, what we really don't know enough about is, does this actually, come about through a change in brain creatine concentrations themselves? And I suppose it's really crucial because developing our knowledge on the kind of interaction between supplementation and brain creatine and these potential benefits, it will give us some more understanding of the actual mechanistic pathways that are underpinning these effects. So that leads us nicely on to actually how creatine is measured in the brain. And there are a couple of different ways of doing this, regardless of which it is performed by conducting spectroscopy using a magnetic resonance scanner, as you can see in the screen. And it depends really on whether you're interested in looking at total creatine, which is the overall pool of creatine, so that's free and phosphoryl creatine, or if you're looking at specifically phosphoryl creatine, as depending on what you want to assess, you'll either be using proton magnetic resonance spectroscopy or MRS, in the case of total creatine, or phosphorus MRS or 31P MRS. And the reason for having to use these different techniques is that it ultimately comes down to the actual nuclei of interest. So with total creatine, you can saturate those hydrogen nuclei and they'll have a distinct resonating frequency that will be able to differentiate total creatine from other metabolites along that spectrum. But what happens is, because phosphocreatine and total creatine within this proton MRS scan resonate at the same frequency, you can't actually differentiate between what's free and what's phosphoryl creatine. And so if you do want to focus specifically on PCR, you need to use a phosphorus MRS, which specifically targets the phosphorus nuclei, that will again give that PCR its distinct characteristic, which will then allow you to capture that metabolite. Either MRS techniques use typically single voxel or a multi-voxel acquisition. So this is essentially just an area within the brain. So you just put a three-dimensional cube, if you like, in the certain region of the brain that you want to assess. And the multi-voxel is more so a grid that you would place in the brain region of interest to acquire that data from. So just to show you what a proton or a 1H MRS spectra look like. So this is what I'm speaking about. This is a single voxel acquisition here from the frontal lobe, you can see. And you can see along that spectra, all these different peaks correspond to a different metabolite. And with proton MRS, creatine resonates at two distinct points along that ppm scale, at around 4 and 3.1. And so it's this distinct resonating frequency of the hydrogen nuclei that are attached to this creatine molecule that will give us the opportunity to actually distinguish between the other metabolites. And then this is a 31P MRS spectra, obtained again from brain tissue. And again, you can see quite clearly in this case the distinguished peak corresponding to phosphocreatine. So how do we process the data then once we acquire it? And so there's several different softwares that are available, Osprey, LC Model, and Tarquin being perhaps the most widely used. And they each have their different underpinning algorithm, as you like. And as we'll speak about a bit later, it does matter what package you use. It will affect the overall output. And so it's quite important to keep the software that you're using quite consistent until we better understand the actual algorithms that are underpinning these softwares. But just for the sake of this presentation, I'm going to bring you through an Osprey workflow. So the first thing you can see there is we would load in our raw water suppressed data. So this is going to give us this spectra that we saw in the previous slides. This is a proton MRS acquisition. And then the second step would be to load in what we call a water unsuppressed acquisition. And this is taken from the exact same region in the exact same voxel that we would take the water suppressed data. But what this allows us to do is actually use this unsuppressed data. So this is essentially just a hydrogen peak. It allows us to use this to actually reference against our water suppressed data. And that will allow us to actually establish an absolute concentration for our metabolite of interest. The next step would then be for the software to run the modeling of our signals. And so you can see the modeling is beginning to separate the peaks from each of the different metabolites. So our metabolites are down along the blue lines here underneath that initial spectra. And this will allow us to start to distinguish between what is each of these metabolites. Another important factor you can see on the top right hand side of the image is our structural data. So you can see the structural data, what we use that for is we use it to try and identify the fractions of tissue in the voxels. And so this is important because when you go and compare data from different individuals or different measurements, you want to make sure that the actual tissue fractions in the voxel are consistent as across those measurements, or else you'd need to correct for that, as that will affect the creatine concentrations. And then the final aspect of this workflow will be the quantification table. And this is essentially going to give you your millimolar concentration for each different metabolites. So if we just move on to the actual reliability of this technique, and this is a really important point. And when we first kind of began to get into this area and we searched the literature, the available literature, quite worryingly, there was quite broad margins of error reported to date. And so combined, these data suggested that the change in brain creatine concentration would need to be between 5 and 20% to actually be distinguishable from measurement error, which is a very broad margin of error. And I think another point of discussion is really the quality of these studies. So they've been very small in nature. They haven't considered multiple brain regions. And one of the biggest aspects that's relevant to this area is that every study included a very long delay between their repeated measurements. And this was up to 10 days. Now, this is a problem because when you're trying to really pinpoint the error band of an actual technique, you need to really try and mitigate any sort of other factors that might be affecting the reliability estimates. And so if you have a delay of 10 days, there's a lot of potential there for actual biological variation to occur in creatine and for other factors to affect the quantification. So you really need to have a very quick turnaround, I suppose, between your repeated measurements to try and actually establish what the measurement error of the technique is. And there was very little data on actually PCR. And I think the largest issue with this is that if we consider the studies that have looked at supplementation with creatine with a view to potentially increase the creatine stores in the brain, at the same time that these studies were published, these reliability data were available, and we can't really be sure what we're seeing here because of the fact that the margins of error were so broad up to now. And so they really did encompass a lot of the supplementation effects that have been reported to date, which broadly range between 3 and 9%. And I suppose the importance of this is really because of the fact that these techniques are going to be used more. There's definitely growing interest in this field, and I expect it to continue to grow in years to come. And so there's going to be an increased demand on these techniques to actually measure brain creatine. So we really do need to actually identify, how we can improve the precision of the measurement before we kind of leapfrog and jump into actually including brain creatine as an outcome. And so this is what we're going to focus on for the next 15, 20 minutes. So what are the potential sources of error when we're measuring brain creatine? So the first one is actually how well we reposition the voxels of interest. And it can be very challenging. So repeatedly positioning the voxels of interest, or that measurement area in the region of brain, can be challenging between people and between time points. The second factor is in relation to the participant, and you kind of have a bit of control with this, but also some of this is out of your hands. But it ultimately, comes down to how still the participant can stay in the scanner, as any movement of their head or even their body might affect the measurement location in the brain. And this will then have a knock-on effect of differences in the tissue fractions of the voxels, and then ultimately the quantification of creatine that you're picking up. The third point is in relation to the acquisition parameters that you set on the scanner, and also the analysis procedures with what analysis package you use. As again, these are going to be things that if you go and change these between people or between repeated measurements, it's going to have an impact on the actual quantification that you can pick up. And another important point is really how you express your data. So a lot of the work with spectroscopy haven't actually reported much on creatine, and that is because creatine historically has been assumed to be quite stable, and so it has actually acted a lot of the time in these studies as the denominator in which other metabolites would be expressed against. And a lot of the spectroscopy studies report the outcomes as this ratio of one metabolite to another, rather than actually calculating the absolute concentrations of these metabolites. As there can be some challenges with actually attaining the right data to be able to calculate these absolute amounts, but it is important in order to improve the overall interpretability of our work. And then the final thing is our physiological variance. So creatine levels in the brain, they are likely to be dynamic, and there are going to be factors that can potentially affect these measurements. And so we really need to kind of improve our understanding and potentially have a better appreciation for what might actually affect the creatine in our brain, and then keep these consistent. So this will be what we speak about towards the end of this presentation, on some acute effects on brain creatine. So then, based upon these error sources, what can we do to try and improve the precision and repeatability? And this is some of the stuff that we worked into our reliability study, which I'll present in just a moment, to try and improve our precision. So the first thing in relation to this voxel localization, the best thing we can do at the minute is use anatomical landmarks within the brain, within the slices, to try and really pinpoint where we're positioning the box or the voxel. And in combination with the actual previous acquisition image, so you can pull that up beside where you're planning your voxel, and to try and really replicate where you had that previous positioning. And there are some automated and semi-automated approaches being developed, which will be really, really useful in this area to try and reduce the margin of error in this reposition, because ultimately there is quite a lot of operator dependent potential error with this, and so again, training the operator, having the same operator use and plan the scans within a specific study is going to be important until we have these automated approaches. In terms of our participant compliance, so as I mentioned, this can be somewhat out of your control, but what you can control is making sure the participants are as comfortable as possible in the scanner. So what we do here in the Institute of Sport is we use quite a lot of different cushioning to place in and around the head within the head coil, just to make sure they're as relaxed within that coil and to mitigate any potential head movement during the scan. And we also offer, even blankets to put over the legs to try and keep the participant in a comfortable temperature during the scan, because it can be quite cool, typically in these MRI rooms. In terms of our acquisition parameters and our analysis packages, what we need to make sure is, until we better understand the effect that these have, is just use the same parameters, same analysis package, and the same settings within those packages for every scan, and for every participant within a certain study. And what's important with this is that if you're interpreting other work, you do need to really have a quite a large appreciation for the specifics underpinning the acquisition, because these will affect the overall outcome. And so it might not be as easy currently with the way the reporting is with some of these studies to actually interpret across different studies using different settings. And then in terms of this physiological variance error source, what we can do at the minute is just try and control our studies and try and design our studies in a way in which we're really controlling as best as possible for these things that may affect creatine, such as time of day or diurnal variation, previous exercise or stress. And if we can't necessarily control for some of these, obviously stress is a very difficult one to control for, but we need to really record these sort of characteristics within our participant population throughout the studies. So based upon these error sources that we identified and these kind of plans to try and address these sources, we really wanted to, before we jumped into any experimental work here in Manchester, we wanted to identify how reliable our proton MRS technique is for measuring creatine concentrations in the human brain. And this work has just recently, just before the new year, been published in Experimental Phys. So if any of the audiences are interested in the finer details that I might not have time to go through, I would direct you towards this paper. So what we did for this study is we recruited a bunch of healthy adults, young healthy adults between 20 and 32 years. Half were female, half were male. We brought them in, we put them onto our 3 Tesla scanner here with our 20-channel head coil. And we selected 3 different brain regions. So we chose the midbrain, the visual cortex, and the frontal cortex. And we set up 2 different protocols. So the first protocol we looked at was an intra-session protocol. And this essentially involved, at the start, we had our localizers, typical localizers, our high resolution structural images. And then we had our voxel of interest placement for our different brain regions. And importantly with this, what we did was we linked the placements between the repeated measures. So once we placed it initially, let's say in the midbrain, that would then be copy referenced into our second acquisition of the midbrain. So we eliminate any operator error here. And then we had our inter-session protocol, which was the more kind of realistic of probably a repeated measure study design, is that we had, again, repeated measurements of the different brain regions, but within or between these repeated measures, we had a complete participant repositioning from the scanner. And importantly, this was very brief. So this was all performed within the space of definitely within 5 minutes. And so this eliminates any potential for the biological variance to creep in. And so then we had our repeated measurements after this reposition. And so this reflects more of what would happen in a typical study. And so we analyzed our data using 2 different software packages with Tarquin and Osprey, because we wanted to kind of get more insight really into how this might affect our measurements depending on what package you use to analyze the data. And then notably, we collected water and suppressed data to be able to then determine absolute concentrations of creatine. So we weren't limited to reporting them as a ratio to another metabolite. And then we did quite a lot of work in terms of how reproducible our placement of voxels were in our inter-session protocol. So for this, we established and calculated the voxel tissue fractions. So how much was cerebrospinal fluid, how much was gray matter, and how much was white matter in these boxes. And we then went a step further and actually established, okay, what's the displacement between the repeated localization of these voxels for each of the brain regions for every participant. So just to show you an example of the voxel placements for each of the brain regions. So you can see in the top row up here is our midbrain. The middle section, middle row is our visual cortex, and then the bottom is the frontal cortex. So you can see the white is just where the voxel or the region of interest was for each of these measurements. And then you can see on the right-hand side of the figure the different voxel fractions, so how much was each of the different tissues. And then you can see there on the right-hand side of the screen the average spectra that we obtained for each of these areas. So for the midbrain, the visual cortex, and the frontal cortex. And you can see total creatine, where we looked at towards the start of this presentation. We can see that it is resonating at these two distinct points along that parts per million scale for every region there. So what did we find with this? Firstly, in terms of our intra-session data, we found very, very good reliability and repeatability. So we found our correlation coefficients and our ICC values were over 0.9. And we found our minimum detectable change being at around 1.2% or less. And I suppose it's what you would expect, really, without repositioning the patient, without repositioning the voxels yourself. So we had no kind of operator-dependent placement of that for the relocalization. And then if we look at our inter-session data, you can see they were quite similar. So you had, again, very high correlation coefficients and ICCs, slightly lower than our intra-session, which you'd expect, but they were still upwards of 0.9 or just over 0.9. And then importantly, we found again this minimum detectable change value of 1.9% or less here. And so very small mean differences between these repeated measures. What we did find, and was quite interesting really, is quite large differences in actually total creatine concentrations across the different regions. So we found differences of up to 24% in total creatine across brain regions. We found the highest in the visual cortex, followed by the midbrain, and then the frontal cortex with the lowest concentration. So this is again is kind of suggesting to us that there may be this metabolic specificity in terms of the concentrations of creatine that are found in these different brain regions. And as you may have kind of thought, looking at these data, although there was very high correlation between the two packages of a 0.9 here, there were actually quite large differences in creatine concentrations determined using Osprey versus Tarquin. So you can see in the graph here, for every brain region, whether it be midbrain, visual cortex, or frontal cortex, there were very large differences in the actual amount of creatine concentrations depending on the package. And it's very difficult to pinpoint what these reasons may be, but just to note that these packages are based upon different algorithms. They're based on different modeling domains, one based upon frequency and then the other based upon time. And there is different, I suppose, steps involved in the actual quantification of creatine using these packages. And I suppose it just really enforces the importance of actually considering the analysis package in the study design, and actually when you're comparing studies as well, how that might influence the overall data that you're looking at. So based upon this initial reliability study, what do we know now about measuring creatine in the brain, and what do we still need to work on? So we know that we can reliably measure creatine in the brain with a very small margin of error, but we do really need to focus on designing our studies meticulously. We need to really focus on the margins or the error sources that I spoke about earlier, and how best to address this within our different centers. And so, although we found quite a small minimum detectable change with our data, this does need to be established in, a center-specific manner before any experimental investigation. So you really need to kind of pinpoint the precision of your technique before your experimental studies. And then the final thing we know now is there is this quite large region-specific and software-specific differences in creatine concentrations in the human brain. And this is, going to have implications for considering what brain regions to assess, considering how we interpret existing data using different packages, perhaps, and actually how we will design our studies moving forward. What we still need to know is whether these findings can be shown in other populations of interest. So these are young healthy adults. And so whether we can get this reliability and precision, I suppose, with older populations, perhaps, or diseased populations who may find it more difficult to stay still in the scanner, may have more dynamic brain creatine concentrations. It remains to be determined, really. And then we need to go and try and further identify what are the factors that we need to be controlling in our study design that may affect creatine, such as diurnal variation, sleep quality, stress, or prior activity. And we'll go on to focus on the exercise component of this in just a moment. And then the final point here is in terms of which analysis package we use. So at the minute, from our work and in our hands at least, we have these two packages that are giving us this high level of precision in our measurement, but actually we don't know, are we having that, or do we have that level of accuracy yet because of the differences between the package. And so I think this is going to really demand some phantom work with the scanner, placing a known concentration of creatine within the MRI scanner, doing our spectroscopy, and then analyzing with these different packages to see which one closely quantifies according to that known concentration in that phantom. So that's the kind of reliability aspect. Again, if there's further questions on this, please reach out and I'll include my contact details at the end of the presentation in terms of optimizing the technique for brain creatine assessment. What we've moved on now in the Institute of Sport here in Manchester is trying to better understand what are the acute factors that might affect brain creatine concentration. So the first one of these that we've chosen to assess really is what are the acute effects of exercise on total brain creatine concentrations in the human brain. Because we know anyway in skeletal muscle that there's definitely this reduction, and the creatine in our skeletal muscle is used quite quickly with these explosive and high intensity periods of exercise. But we really do not have much information on what is shown actually in the human brain. So for this, this is an ongoing project. At the minute, we have 10 adults that have went through this protocol. Again, they're young adults. And what we do this time is we put them through a graded cycle test on a cycle ergometer. And this test lasts approximately 15 minutes. The duration is dependent on how that subject reacts in relation to the heart rate, as we do work these individuals up to a predicted heart rate maximum of 85% and a rate of perceived exertion of 17 and above. So we're definitely getting to that hard level of perceived exertion. We, again, are using our 3 Tesla scanner with the same 20-channel head coil. This time, what we're actually doing is we're incorporating some single voxel spectroscopy, which we would have done in the reliability study that we just discussed in our frontal cortex and visual cortex. But we've also decided to implement some multivoxel chemical shift imaging, which essentially gives us more of a global, if you like, indication as to the change in creatine. So the CSI grid is this top box where you can see the red grid through the midbrain. So you get basically, in our case anyways, an 8x8 grid, and in each of those boxes you get an individual spectra. So it gives us a more an idea as to the global effects of exercise. So in terms of the actual study design, we have the pre-measurements. So we get the person into the scanner, we take our single voxel spectroscopy in the frontal cortex and the visual cortex, and our multivoxel chemical shift imaging in the midbrain. We then get them out of the scanner. They go through this exercise test that we just detailed. And then immediately after the exercise, they're back into the scanner. They have the structural scan taken again, relocalized the voxels, and they have at 15 minutes post-exercise their first single voxel measurement in the frontal cortex and the visual cortex. We then follow this with the multivoxel CSI, and this occurs at approximately 25 minutes post-exercise. And this then again affords us a bit of a gap between our repeated measures of our single voxel work to try and see if there's any transient changes in creatine in the brain. And then at this post 30 mark, we have our second acquisition for our single voxel spectroscopy in the frontal cortex and visual cortex. And I suppose, in an ideal world, you'd continue to take these measurements for a longer period of time, upwards, closer to an hour would have been preferable, I guess. But the reason we didn't include any more repeated measures is because the time burden on the participant at the minute is at around 90 minutes. And so we didn't want to really put too much of a burden on the participants with this. And actually, looking at our data, it doesn't seem to be too much of a factor. So what did we find then? Firstly, let's look at our single voxel spectroscopy and let's look at our frontal cortex. So with this, we found that at the post 15 mark, we found exercise had an acute effect and it was acutely reducing brain creatine. So you can see in this kind of light purple bar that the creatine concentrations at post 15 or 15 minutes post exercise were around 9% lower than what they were at the baseline. And then at the 30-minute mark post exercise, you can see that the creatine concentrations were still significantly lower than baseline at around 5% lower, but you can definitely see this, or a sign at least, of a return to baseline concentrations. So it does seem to be quite an acute effect here in the frontal cortex. In our visual cortex, you can see no change. And this is really what acted as a bit of a control, really, as we wouldn't necessarily expect there to be any change in the visual cortex in response to exercise, whereas in the frontal cortex, we would have imagined there to be, with more of a involvement in controlling or those fine motor movements and our overall movement and exertion during the exercise test. And then for our chemical shift imaging, so again, just to remind the audience, this is the grid through the midbrain, giving us some more global insight in terms of the creatine changes. We found at the post 25-minute mark that there was, again, a significant reduction in the total creatine resonance signals in this case of around 8%. And then on the right-hand side in the darker red, you can see the overall fold change data there at around 0.93. So we found this quite interesting, really. And another important factor that we went and looked at is actually the region-specific changes. So you can see here in this figure, this is essentially a heat map that is looking at the percentage change in creatine concentrations from baseline averaged across the group. And you can see for each of these different boxes, there would have been a single spectra acquired. So as I mentioned, it's an 8x8 grid. So you have 64 different spectra in these boxes, and that allows us to get an insight into region-specific changes that are occurring here. So I suppose it is quite apparent that there is definitely overall a reduction in creatine concentrations, but there are interesting region-specific differences here. And this is definitely going to warrant some future, more in-depth investigation as to what these different regions are, and whether this is found consistently in between individuals. But just quite an interesting finding, I thought, that there is this region-specific response to this acute bout of exercise. So based upon what we have so far, as I mentioned, this is ongoing. We're still recruiting people for this, and we're developing our study as we go. What we know now about the effects of exercise, creatine concentrations do seem to be dynamic in the brain. And again, this is kind of adding to the body of literature that's becoming available in suggesting that we do need to begin to challenge this historic assumption that creatine is really stable in the brain. So it does have the potential to change quite dynamically, according to our data. Brain creatine concentrations do respond acutely to exercise in certain regions. And this again demonstrates that the brain is reliant in part on creatine for energy during high intensity activity. We found that the areas involved in motor function, so our midbrain and our frontal cortex, observed the most marked reduction in creatine concentration, whereas our visual cortex, there was no sign of any change. And I suppose collectively what these data do is that they really do reinforce the need for us to consider external factors, such as exercise, in our standardization protocols for repeated magnetic resonance spectroscopy, looking at brain creatine. So even if these factors aren't our, primary interest, we should consider them in our study design. What we still need to know, we need to find out, this is a acute bout of high intensity exercise on a cycle ergometer. Does exercise modality matter? Are these findings shown in people who undertake low intensity, long duration activity? What effect does sport have? So are these findings shown in sports that involve high intensity exercise? Are they more pronounced, perhaps, if there's also this demand in our cognition and decision making in these sports? And so this can really start to become potentially useful in identifying the demands of different sports or exercise on our brain creatine concentrations, with a view to potentially having more refined nutritional guidelines in and around these periods of activity, with a potentially a pre or post exercise dosage of creatine, with a view to actually help maintain creatine concentrations in the brain, or if in the case of the post-exercise, help to restore the creatine concentrations, and ultimately support our brain health of these athletes or these general population people. And again, we do still have this question. There are a lot of different potential factors that might affect brain creatine, as we mentioned throughout this presentation. So we do need to develop our literature evidence on the different factors that might be important for us to consider when we're assessing creatine. And this is something that we'll be certainly working on here in the Institute of Sport. So just to conclude the presentation with some take-home messages, I suppose, and some future direction. Measuring brain creatine can be done reliably. I suppose the important thing is that we are really critical on our study designs, and we're really tightly regulating our study designs so that we can really focus on minimizing the potential error sources of our technique. And with that, what we need to promote as a scientific community is actually better reporting of, what our analysis parameters are on the scanner, what the quality metrics of the spectra are, because this has a massive effect, what the signal to noise ratio is, what the water full width half maxima are, that these really important quality metrics need to be reported so we can actually critically evaluate the literature that's being published. And this will help to actually develop our understanding of this area in general, really. Creatine concentrations are dynamic in the brain. And there are, accumulating data to show that the historic assumption that they're stable should perhaps be challenged somewhat in an appropriate manner. And we need to begin to understand what could potentially affect our concentrations of creatine in the brain. And this is important because, this work is, it may not be what we're necessarily primarily interested in, these factors, diurnal variation, sleep, exercise, but they may be really important to understand how they might affect our actual quantification of creatine, because we need to control for these if they do indeed have an effect. And then, as we kind of touched on at the start of the talk, in terms of, why are we measuring brain creatine, and, why are we trying to improve the brain creatine dynamics? And I suppose it does have quite promising applications across the public health and sport environments. And as we mentioned, there's certainly plausible relevance to any of these scenarios that have a very high energy demand and or have a impairment to the typical energy metabolism in our brain. And going back a bit, we need to, re-establish using these better designed studies and lower margin of error with our technique. We need to establish what the effects of creatine supplementation and how efficacious that is in actually increasing brain creatine concentrations. And importantly, what is the optimal dosage for doing this? There are zero dose response studies that are available to really show us how much should we be supplementing with, how long should the supplementation be, and what are the effects on the actual concentrations of creatine in the brain. So we really need to start develop this body of evidence as has been developed for skeletal muscle. And then ultimately, it comes down to, if we do observe these increases in brain creatine, is this the potential explanation for the benefits that we are seeing currently with creatine supplementation on several different brain health and cognitive function outcomes? So this will really bring us some, shed some light on the mechanistic pathways that these health benefits may be brought about through oral supplementation of creatine. And then the final point is, to stress that if it is possible within your labs, if you have the possibility of having a multinuclei spectroscopy setup focusing on our proton or our 1H MRS and our 31P or our phosphorus MRS, these are ultimately needed to be able to provide the full picture of brain creatine dynamics, so free creatine, phosphoryl creatine, and the overall creatine pool, and how they relate to oral supplementation of creatine and our cognitive health or our performance outcomes. So we really need to start building really high quality body of evidence using this multinuclei approach to better understand the full picture here. And with that, I'd like to thank you for your attention. And again, apologies I can't be there in person, but please do reach out to me with my email or my Twitter handle if you have any questions about anything that we spoke about. So thank you very much.